Okay, I think it's time to get started. We want to stay on time for the day. So it's my pleasure to introduce the Basic Science Plenary Session speaker today. Dr. Nicolas Chomont is an associate professor in the Department of Microbiology, Infectious Diseases, and Immunology at the Université de Montréal, as well as a researcher at the Chomont Research Center. He earned his PhD from the University of Paris in 2004, where his research focused on the cellular mechanisms underlying HIV transmission. During his postdoctoral work, Dr. Chomont identified the CD4 T-cell subsets responsible for the persistence of HIV in individuals on antiretroviral therapy. Currently, he leads studies aimed at elucidating the immunological mechanisms that drive HIV persistence and developing innovative therapeutic strategies to eradicate the virus. So join me in welcoming Dr. Chomont for his talk.
Thank you, Amy. Thanks very much for the kind introduction. It's really a pleasure to be here. So today I will talk about the future and about curing HIV. And as you will see, it's not only the future, it's actually already a reality. Some people, as you may know, have already been cured of HIV. These are my conflicts and the learning objectives for today. So we'll try to understand why we do not have a cure, I would say a cure for everyone yet for HIV infection. Then we will explore how the viral reservoir is established, how we can identify the tissues and the cell types in which the virus persists in people receiving antiretroviral therapy. And finally, I'll discuss some of the new approaches that have been designed to try to cure HIV infection. So the first question I'd like to address, which is not an easy one, is what is a cure? And actually, this is a question you should ask all the people you know who are living with HIV, and you might actually be surprised by the fact that there is a lot of diversity in the responses you will get. And this is not very surprising because even if you ask yourself, how do you envision a cure? What is your definition of a cure? Well, you may say that you'll be cured when you won't need to take HIV medication. You may say that a cure is that you will not be stigmatized or criminalized anymore. A cure is that you will not transmit HIV, that you will be free of HIV, that you won't have comorbidities, that you will have a normal quality of life. And for many people, it's actually all of the above. So that's actually a pretty high bar if you'd like to develop a cure for HIV because you'd like to solve or to resolve all these associated issues. Why do we need a cure? It's funny because I still need to include this slide in my talks. I thought that everybody agreed on the fact that we need a cure for HIV infection, but I realized that some people actually are not convinced of this. There are several reasons why we need a cure. First, because the problem of stigmatization and criminalization is not solved, and particularly here in Canada, and we were discussing this yesterday. Access to antiretroviral therapy for everyone is a formidable challenge, still now, and particularly in this context where PEPFAR is cut by the US government. There is toxicity associated with some of the antiretroviral drugs we're using in people living with HIV. There is persistent inflammation and comorbidities in people living with HIV. And finally, there is an economic burden of HIV and antiretroviral therapy that has some weight on our healthcare systems. This is a slide I like very much that actually has been shown to me first by Madeleine Durand, in which you're looking at the life expectancy of people living with HIV. And as you can see, during the past 30 years, the life expectancy of people with HIV has greatly improved, and actually it's getting very close to the one of people who are not living with HIV. But at the same time, if you look at the rate or the frequency of people who are comorbidity-free, you'll see that there is still this huge gap between people living with HIV at the very bottom of the graphic and people who don't have HIV. So people with HIV still develop much more comorbidities than people who do not have HIV, and this is still true in the era of modern antiretroviral therapy. So we still have a lot of progress to make in this area, and that's another reason why we need to develop a cure for HIV infection. Another reason why we want to develop a cure are these appalling statistics that we get from the Public Health Agency of Canada, with an increase in the rates of new HIV diagnoses all across the country and particularly in the prairies. More than ever, we need a vaccine or a cure to limit the epidemic of HIV in Canada and worldwide. Why don't we have a cure? Well, we don't have a cure because antiretroviral therapy does not eradicate the virus. So if you look at people who are taking ART, usually ART works very well, right? So your viral load will go down in a matter of weeks and will become what we call undetectable, at least below the levels of detection of what we use in the clinics or at the hospital. But the virus is still there because if these people stop antiretroviral therapy, you will see a viral rebound. So really this indicates that during this phase, the virus is still somewhere, still persisting somewhere in the body and can cause rebound when the therapeutic pressure is removed. So our job and the job of many people working on the development of an HIV cure is try to identify where HIV hides in people living with HIV on antiretroviral therapy and try to develop new strategies to get rid of what we call viral reservoirs. A slide on the concept of HIV latency. So you've probably heard this word many times. What's HIV latency and what's a viral reservoir? So HIV latency is something very peculiar, which is actually not the majority of the scenario in which HIV infects a cell. So usually when HIV infects a cell, it likes to infect what we call activated CD4 T cells, which in other words are cells that are pretty much excited. And when the virus gets into these cells, it replicates actively, produces new viral particles, and this leads to the death of the infected cells. But on very rare occasions, I would say, resting CD4 T cells can get infected. And when that's the case, these resting CD4 T cells don't die. They actually survive. And the virus within these cells can actually survive for the lifetime of the cell as well. So HIV has the ability to remain silent, or we say also dormant or latent, in a very few cells. So this is usually a property of viruses like herpes viruses, like CMV, herpes viruses, EBV, VZV have the ability to do that. And HIV is not an herpes virus. So the way HIV establishes latency is still a mystery. It takes advantage of some particular T cells that are memory resting CD4 T cells, and this is quite unique to HIV. But the result is the same, is that the virus is able to establish this persistent infection that will persist for the lifetime of the people living with HIV. Importantly, another important definition of latency is that the virus has to be reactivated or has the ability to be reactivated at one point. And this is true also for HIV. So these resting CD4 T cells can persist for many, many years. But upon reactivation, they have the ability to produce new viral particles. So if I go back to my previous slide, and now we are adding a little bit of biology in this. As you can see, the viral load is caused by cells that are actively replicating the virus, so these activated CD4 T cells. And antiretroviral therapy has the ability to block new infections, so this pool of red cell will go down very rapidly with antiretroviral therapy. However, the resting CD4 T cells, the latently infected CD4 T cells, are not affected by antiretroviral therapy. So they persist for years in people on antiretroviral therapy, and as I said just before, when they are reactivated, they can give rise to a new peak of viremia, and this is the reason why people rebound after antiretroviral therapy interruption. So again, it's very important to understand what are these cells, where they hide, how can we target them. Our goal is to develop a cure, which means we want people living with HIV to stop antiretroviral therapy without developing experiencing viral rebound. And for that, again, we need to better understand the biology of these latently infected cells and try to eliminate them. One of the main issues we are having with this is that this HIV reservoir, so this pool of blue cells, the latently infected cells, is extremely stable. And this is a study from the group of Bob Silicano and others in the US who have looked at the dynamic of the HIV reservoir after decades of antiretroviral therapy. As you know, ART has been, or triple ART, I would say, has been developed in 1995 and 1996. So now we have some ways to actually measure the half-life of the reservoir during almost 30 years. And as you can see here on this slide, this HIV reservoir, which has been measured by a particular assay, which is called the VOA, which is indicated here, this reservoir is so stable that even 100 years of continuous ART would not be enough to eradicate it. So clearly, we need to do more than antiretroviral therapy. We need to target this reservoir by designing innovative, creative eradication strategies to get rid of the HIV reservoir. An important question that I mentioned already several times here is, where does HIV hide? And this is not an easy task to answer this question, because the answer is pretty much everywhere. We've learned this from the work from Sarah Gianella and Antoine Chaillon at UCSD, that HIV can persist in many deep tissues. And even though these first studies were actually very informative on the existence and the location of these HIV anatomical reservoirs, there were still a lot of missing informations on how the virus can persist in these tissues. And so with the help of CAHR, which is a group of scientists in Canada funded through a CAHR program, during the past 10 years, this CAHR is an initiative that was created by Eric Cohen, and I'm now the new lead of CAHR, and I'm very grateful to be working with all these fantastic scientists across Canada. We had access through CAHR to tissues, autopsy tissues, from 2 participants who very generously gave their body for research on HIV reservoir. The first participant was from Ottawa, with the help of Jonathan Angel, who's been working on this case, and the other one from Edmonton, with the help of Chris Power. And Cecilia Kosteniuk is now the new lead on this end-of-life study. The story of these 2 participants are very similar. They were both in their 60s. The first one died because he asked for medical-assisted death. The other one passed away from a B-cell lymphoma. They were both diagnosed during their 80s, and you can see their history is quite complicated with lots of comorbidities, particularly for the Edmonton participant. So again, thanks to these 2 participants, we were able to collect many tissues to really address the question of where does HIV hide at the global level. And this is the list of tissues we collected with the help of many people in many different labs across Canada, because you can imagine that processing all these tissues was actually quite challenging. So with Aligé Nabian, Petronella Ancuta, and many others, we processed these tissues and analyzed them for the presence of HIV reservoir. Collected a lot of lymph nodes, and the reason for this is because CD4 T cells, the main reservoir for HIV, love lymph nodes. CD4 T cells are lymphoid cells. They reside in the lymphoid tissue. And this is the reason why we had a particular focus on lymph nodes, so mediastinal, axillary, hilar, but also the spleen, the mesenteric lymph node, the inguinal lymph node. And also less conventional reservoir, we say, such as the testes, the brain, the lungs, the liver, a different part of the gut, from the duodenum, jejunum, ileum, colon, and rectum. And the first thing we did is a simple PCR for HIV DNA. So remember, HIV is a retrovirus, so it's an RNA virus in the plasma, but when it gets into the cell, it's in the form of DNA, and the form of HIV persistence is HIV DNA. So if you want to measure the reservoir, you need to measure HIV DNA. And we measured HIV DNA in several tissues from these 2 participants, on the left from the Ottawa participant, on the right from the Edmonton. And you can see the list of tissues again. LN is for lymph node, and we have several dots in each column because we repeated these measures. I mean, we're dealing here with humans, not with mice, so you can imagine that when you get a human liver, it's huge, so you want to make sure that you repeat the measures in different pieces of the organ, and this is what we did. As you can see, HIV DNA was detected in all tissues we analyzed, with the highest frequencies found in the lymph node, in the spleen, in the liver, and in the lung. Something important when we measure HIV reservoir, for many, many years, we've used this simple HIV DNA PCR. So basically trying to detect a small piece of the HIV genome, and this PCR worked very well. And if I show you what they do, it's here in pink. So typically, when you do a PCR for the GAG gene, you're trying to capture this small portion of the HIV genome. So this is the HIV genome in its entirety, I would say. And these are different sequences that you would get if you try to do the full genome sequencing in one participant. So in green are genomes that are completely intact, but as you can see, most of the genomes persisting in people on antiretroviral therapy have defects. And the reason why they have these genetic defects is because HIV is making a lot of mistakes when it's replicating. So the important message here is that when you do only a PCR for the GAG gene, you're capturing all these genomes that are positive, including those guys, for instance, which are orange, so clearly defective. They still have this small portion of the GAG gene, but they have other defects. So you would classify this as an HIV reservoir, but it's not, because there is no way this virus will replicate. So if you really want to measure the real reservoir, the one that is able to induce viral rebound, it's very important to actually sequence the complete viral genome, or to get a sense or an estimation of the size of these genetically intact genomes. And this is what we did, or what Caroline Dufour did in my lab during her PhD. She actually generated 300 sequences of near full-length genomes from the Ottawa participants. So again, at the top here, you're seeing the HIV genome in its entirety, from the 5' LTR to the 3' LTR. You recognize all the genes, GAG, POL, and ONV, and all the other genes. And you can see there are many of these genomes that have like huge deletions that are clearly not intact and probably irrelevant for cure studies. And actually, if you classify all these genomes according to their integrity level, you would see that only 1.7% here at the bottom of the slide in light green are genetically intact. So that means that 98% of all the genomes that would be captured by a small, short PCR are actually defective and irrelevant for HIV cure studies. These intact genomes that we found in these participants were found mainly in the spleen, in the mediastinal, and the mesenteric lymph nodes, so 3 lymphoid organs. And we think that most of the HIV reservoir, the true HIV reservoir, is persisting in this lymphoid tissue, not excluding the possibility that other organs, of course, also contribute to HIV persistence, including the brain, the lung, and others. In the second participant from Edmonton, the story was a little bit different. Remember, this participant passed away from B-cell lymphoma, and we believe this condition actually created something, a huge disturbance in the HIV reservoir. We find a 25% of intact genomes in these participants, which is huge. We've never seen that in anyone before. So we are not exactly sure how to interpret this data, but the reality is that we were able to find intact proviruses in all the tissues we analyzed, with the exception of the inguinal lymph node and the duodenum. We will pursue this effort within the CAHR 3.0 team, again, a team of Canadians, scientists, but also, very importantly, a CAB led by Shari Margolis, who is our principal knowledge user, 6 members of our community advisory board. As you can see, we have both virologists, immunologists, and clinicians, plus the collaboration of CADI and CAN for this new CAHR initiative that is just starting now. If you need more information, or if you're interested, please contact me. We always need more input and more people on our team. CAHR 3.0 will focus on the tissue, because we really believe that for HIV cure, tissue is the issue. I put here the 3 teams that will be developed under CAHR. I'm not going to go through the details here on what we will do here with CAHR 3.0. The important part is probably in the center of this slide. Thanks to this collaboration with all our members, we have access to tissues from people living with HIV through the end-of-life study that I just discussed, but also through other protocols with the collaboration of Jean-Pierre Routier and others to collect colon, lymph node, and bone marrow, but also bronchoalveolar lavage, which is a work that Cecilia Kostyniuk and Aligé Nabian are leading. And also we have access to animal models such as non-human primates with Jérôme Estaquier, and humanized mice with Eric Cohen and Elie Haddad. Okay, what would a cure look like? Well, I wish I knew, but we believe that what will be important is to prevent viral rebound. And to prevent viral rebound, you need to target the reservoir. So the idea is to have an intervention before antiretroviral therapy interruption to target the reservoir, decrease its size, and kill the latently infected cells that are the cause of viral rebound. You can imagine that some interventions will lead to control, others will lead to remission, others will lead maybe to eradication. And these are the different eradication strategies that have been proposed so far. The first one is to try to limit the reservoir establishment, and I'll get back to this in a minute, with early antiretroviral therapy. You can also try to reduce the size of the reservoir, and there are several strategies that have been tested, from purging the reservoir, like what we call the shock and kill, with the idea is to wake up the sleeping virus to make it visible to the immune system, to deplete infected cells, to protect cells from HIV infection, or to block the reservoir through a block and lock strategy. We are more and more convinced that eradicating the virus will need not only to reduce the reservoir, but also to boost the immune system by an immunological control of the reservoir. So this can be achieved with a therapeutic vaccine, with immunotherapy, with monoclonal antibodies, and with CAR T cells. CAHR is involved in many of these aspects, and they are listed here with the CAHR logo. Cure is possible, and we know that from at least 9 cases of people who have been cured for HIV. The first one, as you may know, was Tim Brown, also known as the Berlin patient, who was the first person cured of HIV in 2009. Since then, we had 8 additional people who are now considered cured of HIV. The most recent ones were presented at CROI last year, the Chicago participant and the Oslo one. Interestingly enough, only one woman, the New York patient, who has been cured of HIV so far through a cord blood transplant. All these people received stem cell transplant because they needed it to cure their cancer. Stem cell transplantation is still a very risky procedure, which is associated with a 30% death risk, so clearly not a cure for everyone, so we need to do better than this. Some people have the ability to naturally control, but more than control, to naturally eliminate HIV entirely, and this has been seen at least 3 times, one in the Australian patient, one in Lauren Willenberg from San Francisco, and the last one is the Esperanza patient from Argentina. All these 3 participants have no detectable reservoir. Even if you look very hardly, you won't find a single cell with HIV DNA persisting in these people, indicating that on some rare occasion, it is possible that your immune system can get rid of HIV by itself. The most common cases of remission are those that are due to early treatment. And I will not detail everything here because there are many cases that have been described during the past 12 years now. The first ones were the Visconti participants in France, described by Azir Saisserian and others, people who started out quite early on in infection, and after a couple of years on antiretroviral therapy were able to control naturally HIV replication after antiretroviral therapy cessation. Something that I'd like to highlight here in red is that many of these natural remission cases are actually children. This is true for the Mississippi baby, the French teenager, and many others, and I'll get back to this in a minute. With the collaboration of Jintanat and Anne Voronich in Thailand and Lydie Trotman at the Military HIV Research Program, we've been working on this study of acute HIV infection in Thailand for more than 15 years now. This is a fantastic study, and really this is due to the hard work of many people in Thailand, the nurse, the doctors, and most importantly, the participants and their dedication to this study. The idea is to diagnose people at the earliest stage of HIV infection, and as soon as they are diagnosed, to start antiretroviral therapy. And the hypothesis was that if you start antiretroviral therapy very early on, you might prevent the establishment of this long-lived and persistent reservoir. So this is the protocol as it was created, and through this protocol, close than 1,000 people have been recruited so far, and started antiretroviral therapy the day after diagnosis. Louise Lair, who was a master student in the lab, actually used these samples to measure the reservoir, again using the HIV DNA, as we did in the past. And as you can see in this slide, so you're going from the left to the right, PHIV are different stages of HIV infection. So PHIV1 is the earliest stage. That means that you have detectable HIV RNA in your blood, but you're negative for all other markers. PHIV2, you were seeing P24 protein. PHIV3, the first antibodies, and so on and so forth. And finally, the chronically infected people. or people living with HIV at the chronic stage. And so this is very early. People have started out very early on, a little bit less early, and people started out during chronic infection. And we are looking at the size of the reservoir after 2 years of antiretroviral therapy. And as you can see, the earlier you start, the smaller the reservoir will be. And this is true in the blood, but also in tissues. Because again, these participants are so dedicated, we had access to lymph node and colon to measure the reservoir in these tissues. And as you can see, early ART really prevents the establishment of this persistent reservoir, not only in the blood, but also in the tissues. So that's great news, and it seems that early ART works to prevent, at least partially, the establishment of the long-lived reservoir. As I said before, when you're using this short PCR for GAG or any gene in the HIV genome, you will capture only a small portion of the genome. So it's important to perform near-full genome sequencing. And this smiling lady here is Caroline Dufour, who was a PhD student in the lab and who performed near-full genome sequencing in the blood of these participants with the help of Pierre Gantner. And so we generated these full genome sequences and used PacBio sequencing. We are now using nanopore. The point is that we're using these long-read sequencing methods that allow us to get the full genome of these people. And we compare the HIV genome at week 0, so the time the people started antiretroviral therapy, and 2 years later. So the question we were asking here is whether an intact HIV reservoir could be established early on and persist for 2 years on antiretroviral therapy. And so this is the work that Pierre Gantner did and that we published 2 years ago. And the answer, unfortunately, is yes. We found in these 2 participants in green, so these are intact genomes, that were detected early on at week 0 and persisted for at least 2 years on antiretroviral therapy, so still present at week 96. This is true here, and this is true here, clearly showing that intact proviruses can be archived early on in infection, and they can persist at least for 2 years during antiretroviral therapy. And the consequence of this is that early ART is not enough. And when we performed this study in collaboration with Don Colby and others back in 2018, we decided to interrupt antiretroviral therapy in 8 participants who started ART at the FIBIG1 stage, so the early stage of HIV infection. And unfortunately, all these participants rebounded. They restarted ART just after. They're fine. The size of the reservoir actually did not increase. But I mean, that was really the best proof that even though early ART can prevent some of the reservoir establishment, it's clearly not enough to cure or to prevent the HIV reservoir. Something might be a little bit different in children. And this is a new study that we have not published yet that is done in collaboration with, again, Thailand, with Tanya Wiputanakit, who is the principal investigator on this study, with the help of Marta Massanelia, who was a postdoc in my lab for several years. And in these children, we'll measure the HIV DNA prospectively. These children were born from mother living with HIV and have been followed for almost 10 years now. And we have been very lucky to have access to these samples of these children over the past 10 years. So again, in children who acquired HIV perinatally and initiated ART early, these children started out on an average median time of 11 weeks after birth. The pool of cells harboring HIV DNA remain relatively stable after years of ART. Not very surprisingly, this is what we're seeing in many adults. When we use another method of HIV reservoir measure, which is called TILDA, which is measuring something more functional, I would say, because we're measuring the HIV genomes that have the ability to produce viral RNA, you see that it's not very clear, but it seems to be, there seems to be a decay in the size of the reservoir in these children. So this inducible reservoir or functional reservoir might be continuously cleared. And so to really try to get a better picture of this, we decided to perform near-full genome sequencing, which is what we do in the lab now. And I'm showing you here the example of one of this child that has been followed for more than 7 years. So this is the profile landscape of this kid just after birth, just before he started antiretroviral therapy. And you can see lots of intact genomes in green, and actually probably 30 to 50% of intact genome, which is a lot. And we followed this child over many years on ART. This is what happened after 2 years. you see much less intact HIV genomes. After 3 years, even less. And after 7 years on ART, we were unable to detect any intact HIV genome in this child, which is very encouraging, I would say. But this is one example. So what's happening when you look at more children? And this is the summary of what we observed in the entire cohort. In the middle of the pie chart are the number of sequences we obtained. At the top are the number of children we looked at. So you can see before ART, we had access to samples from 3 children. And this is what you would see after 2 years, 3 years, 5, 6 years, and more than 7 years. After more than 7 years on ART, we looked at samples from 7 children, and we detect a single of these participants, a single intact genome. So indicating that in 6 of these kids, we were actually not able to find any intact genomes, suggesting that maybe they naturally got rid of their viral reservoir, pretty much like Lauren Willenberg or the Esperanza patient. So, of course, we will not interrupt ART right now. They are still 10 years old only, so we really want them to still participate in this study to be able to follow them, and hopefully this is what we will do. So again, intact HIV genomes are exceedingly rare after 3 years of ART in this particular cohort. Okay, in the last part of my talk, I'd like to speak about the biology of the HIV reservoir. As I said before, the big issue we're having, there are 2 big issues. The first one is that most of the HIV reservoir is in tissue, which are difficult to access. And the other big issue is that we are not exactly sure what types of cells are a long-lived reservoir for HIV. So which cells should we target to really get rid of the HIV reservoir? I showed you before that the HIV reservoir are smaller in people who initiated ART early. The reservoir may decay faster in children. However, in most individuals, very early ART does not prevent the establishment and the persistence of the HIV reservoir that leads to viral rebound after ART interruption. I should also mention that still today, the majority of people who are newly diagnosed with HIV are not in acute infection. So initiating ART at the early stage of HIV infection is not something feasible on the large scale. So we need to identify the rare cells carrying intact and replication-competent proviruses from which HIV rebound during ART interruption. This is no full slide that shows a lot of data that have been generated during the past 30 years on trying to identify the cells in which HIV persists in people on antiretroviral therapy. Most of the previous studies used HIV DNA as a readout, which largely overestimates the size of what we call the real reservoir. As I said before, if you really want to measure the reservoir, you need to perform near full genome sequencing. There are novel approaches that have been developed to get the phenotype, so to define the nature of the cells in which the virus hides, such as PHIV, developed by Matthias Lichtenfeld, and also the ASAP-seq from Mike Betts. Yachi Ho also has developed novel ways to analyze the reservoir. In my lab, we decided to go with a different approach, which we call the NFL HIV flow. So briefly, what we are convinced, or what we try to convince ourselves maybe, is that it's important not only to look at the genetic intactness of the HIV reservoir, but also at the functionality of this reservoir. If the provirus, so if the HIV genome is integrated in a region of your chromosome that will never get reactivated, it's probably not clinically relevant. So you need an intact genome, but an intact genome that can be reactivated to produce new viral particles. And so we didn't have an assay that could actually capture these 2 parameters simultaneously. And with the help of Caroline Dufour again and Marion Pardonce and others in the lab, we developed this new assay. So this is our approach. We're starting from blood from participants on antiretroviral therapy. We isolate CD4 T cells, and we reactivate the reservoir. So basically, we stimulate these cells with PMA and ionomycin for 24 hours, which wakes up the virus. So the virus is waking up, it's producing viral particles and viral proteins, such as P24, and we developed an assay which is called HIV flow, in which we co-stain P24 intracellularly with 2 antibodies to get a better definition of the positive signal. And then we did index cell sorting, so that means that each of these individual P24 positive cells is sorted in an individual 96-well plate. And then we perform near full genome sequencing on each cell. So basically, we're asking in this well, is the HIV genome intact or not intact by performing this assay? And because we're doing cell sorting, at the same time, we can record the phenotype of each individual cell. So at the end of the day, you're able to associate a given phenotype with the genetic intactness of the HIV provirus, which is the question we were asking. So you're looking at the phenotype of the cells that have intact and inducible HIV genomes. And this is what we found. At the very beginning, pretty much nothing. So there was no difference between any CD40 cells and the CD40 cells that harbor the intact HIV genomes. So on this slide, you can see here, we're comparing the level of expression, for instance, of PD-1, which is an exhaustion marker, on cells that have different type, they harbor HIV proviruses, but proviruses that are either intact, here in green, or that have different types of defects, and we merged all the defects here on the right. And so if you look at the level of expression of PD-1 here, you'll see that it's not difference between cells that harbor intact genomes and cells that harbor genomes with genetic defects. But hopefully and thankfully, we found something that was different. So when we looked at the level of expression of these alpha-4 and beta-1 molecules here, we found that cells with intact genomes, here in green, express higher levels of beta-1 compared to the cells that harbor proviruses with large deletions. And for alpha-4, the cells that have intact genomes express higher levels of alpha-4 than cells that have proviruses with large deletions, other type of defects, or all of them merged together. Interestingly enough, alpha-4 and beta-1 actually work together, and they form an adhesion molecule that is called VLA4. So we are quite excited about this. So it seems that VLA4 might actually be a new marker for HIV reservoirs, and we actually validated this in another assay, which is called the viral outgrowth assay. We sorted memory CD40 cells expressing VLA4 or not, and compare their content in replication-competent HIV. And as you can see here on this slide, we found a 27-fold increase in the frequency of cells with replication-competent HIV in cells that express VLA4. So this was quite exciting, and the good news is that there's an antibody to VLA4. So you might want to target this. It's called natalizumab. It's a humanized monoclonal antibody that targets VLA4. It's approved for multiple sclerosis and Crohn's disease. However, it increases the risk of PML, so clearly we're not going to do this to our participants with people with HIV. The good news is that there are novel generations of VLA4 antagonists that we could use that are listed here on this slide. And actually, the group of Ken Williams already used this natalizumab antibody more than 10 years ago in non-human primates. I'm not going to go through the detail of this study, but interestingly enough, they were able to show that early treatment with this antibody can stop productive infection of the brain and the gut and prevent the establishment of HIV reservoirs in these tissues. Whether natalizumab will have any activity on an already established reservoir, we don't know, but this is something we'd like to find out. I'll finish with a new study from the lab that has not been published yet, which is led by Hélène Roux, a postdoctoral student. And in this study, we're using the same approach, so the NFL HIV flow, but we included more markers. We enrolled 11 people living with HIV, unfortunately all males at this stage. They've been followed up for 4 to 25 years on antiretroviral therapy. And the idea was to get or to determine whether there is an evolution in the phenotype of the HIV reservoir over years of art. Something, and I don't know how many immunologists are in the room, but we found that the bottom line is this. The reservoir cells, the cells in which HIV hides, are very weird. They have a very peculiar phenotype that we don't see in any other CD40 cells. This is true when you look at their ability to produce cytokines, and here you're comparing the frequency of total CD40 cells producing gamma interferon versus the frequency of P24 positive cells producing interferon gamma. Same thing for TNF-alpha, IL-2, and IL-17. And as you can see, the P24 positive cells, so the HIV reservoir cells, they make tons of these cytokines. And actually, they are making multiple cytokines as well. That's shown here on this pie chart. But this is not the only weird thing about these HIV reservoir cells. They express Tibet, which is a lineage marker for Th1, but also a little bit of BCL6, which is a lineage marker for TFH cells. Not much of GATA3, Th2, and FOXP3, Treg. But that's in French. On the other side, surprisingly, they express several of these markers simultaneously. If we look at the cytotoxicity capability of these cells or capacity of these cells, again, this is something that has been shown before by Yachio and others. They're making granzyme A. Granzyme is usually something that is made by CD8 T cells and NK cells, but not much by CD4 T cells. But the HIV-infected cells, again, very weird cells. They are making cytotoxic factors such as granzyme A, a little bit of B, and a little bit of K. And finally, when you look at senescence markers, they express KLRG1 and not much CD57. So this is a summary of these. These P24 positive cells are weird. They express multiple cytokines, transcription factors, as well as granzyme and KLRG1, and immune checkpoints, and BCL2, but I didn't show this data. And we think that this is really a signature that is reminiscent of a senescence-like phenotype. And because of this, we are partnering with Adam Spivak and others to actually test whether drugs that specifically kill senescent T cells, which are called senolytic drugs, could have a positive impact by depleting the HIV reservoir. So for now, we'll do that ex vivo in test tubes. The plan is to move to animal models and eventually in humans, because many of these drugs are actually already approved to be used in humans. So we are very excited about this. This is my conclusion slide. So I hope I showed you that early out can limit the establishment of the latent reservoir, that HIV persists in many tissues, with lymphoid tissues enriched in intact genomes. CD4 T cells express VLA4, which I think is a new marker of the HIV reservoir that we should probably try to look at more carefully. And finally, the reservoir is enriched in senescent CD4 T cells, suggesting that senolytic drugs may be used to reduce the reservoir. Most importantly, I'd like to acknowledge all the people who worked on this study, the people from my lab, Caroline Dufour, Pierre Gantner, Martin Massanelia, Rémi Fromentin, and many others. At University of Montreal, we have many partners and collaborators who have helped us with these studies. Importantly, our collaborators in Thailand, both for the adult acute HIV infection study, but also for the baby study. Our funders, all the CAHR members, who are, many of those are in this room, so thank you CAHR as well for funding CAHR. The study participants, obviously there is nothing we can do without the commitment and the dedication of the study participants. You know that we need to get tissues to perform all these studies. This is quite difficult, and we are very lucky to have some very generous participants. I'd like to finish by saying a word on the situation of the funding by the NIH. So yesterday, we received the appalling news that the NIH will cut any subaward to foreign institutions. So that means that for us in Canada, and I guess that many of you in the room are also getting funds from the NIH, many of us won't be able to continue the work we've been started many, many years ago. Cutting these funds by the NIH means that many people that you see smiling in this picture will lose their job. Among these people are people who have been working with me for more than 10 years because they're research assistants, research associates, but there are also PhD students, master students, postdocs coming from foreign countries who have been supported by the NIH for 1, 2, 3 years. And today or tomorrow or Monday, I will have to be back in the lab and tell them, well, the US government and the new administration has decided that the work you're doing should not be funded anymore. The reason I'm telling you this is because we need to fight. We are not the US, we are Canada, so we need to talk to our government, both at the province, at the federal level, but also to CAHR, to all the funders, to all the partners and the pharma companies and elsewhere. We need to find a way to keep these people with us. This is the future of the science, not only for HIV, but the future of the science in all areas. So it's very important that we support these people we need money to pay their salary after the NIH cuts. So I'm counting on all of you to actually support this initiative and help us in trying to find alternative strategies to pursue the good research we're trying to do. Thank you very much.
Totally agree with that sentiment. Thank you for an excellent talk. It was informative, and most importantly, it was also very accessible. So I think the audience appreciated it. We have a minute left before we're into time for the epidemiology talk. So if there's one quick question, somebody wants to run up to a microphone. Otherwise, we'll let the epidemiology people get set up.
Question?
Sorry.
I'm over here.
Hi, okay.
Thanks. Jessica Prodger from Western University. That was an amazing talk. Thank you so much. And I also really appreciate your comments about fighting the NIH. I have a few students from Uganda who started PhDs this year who are also funded on subawards. So my question is, I mean, the phenotype of the P24 positive cells is really so interesting. I think that's amazing. And we actually have some old data from a cohort in Uganda showing the same thing with the granzyme positive A cells that we have never published. We had a hard time interpreting it. And I think in your much larger, more comprehensive phenotyping makes me feel like, oh, maybe that was like a true signal that we were seeing. I'm wondering, did the cohort that you were examining in that include both males and females? And were you able to disaggregate based on sex?
Thank you, Jess. Great question. So no, unfortunately, the 11 participants to this study are all males. This is not good. I realize that, and this is something we're trying to fix. So for now, I can't answer your question. We are very much interested in difference between men's and women's, actually, so this is something we plan to do.
Thank you. And please accept this gift
Thank you so much.
from CAHR for giving a great talk.
Thank you.